There we go, take the front seats. I always say, if you go to a concert, you pay a lot of money for the front seats. They're free here. You don't pay any extra. Definitely. Did you get my pictures, Ben? No. Oh, I emailed you pictures. Yeah. I was there for the celebration and I didn't know you were gonna be there. No, no. Yeah, I'll be back. Hey, everybody, we'll get started. I originally got to know Matt, on a request from my brother, who's the executive vice president of the American Amusement Machine Association. My brother asked me to give a talk to business owners that owned arcades, bowling alleys, trampoline parks, and also restaurants. They wanted to understand, what is this new virus, this COVID virus, and how can we protect our businesses? I mean, they were getting slammed when the virus first got out there. My brother Pete was the person that they went to find information about how they could protect themselves and what to expect with what was going on. And I thought, what a great, stable gentleman here. I couldn't make the second day, and so I asked Matt to do it, and he did a great job from what my brother Peter said. So that's how I got to get to know Matt a little bit better. Matt works on the features and mechanisms of bacterial stress responses, in particular in Bacillus subtilis. It's a classic microbial model. He also works on the metabolism behavior of Pseudomonas aeruginosa, which is an opportunistic pathogen, and with a special focus on its ability to produce pyrosins, which are phage tail-like intraspecific killing complexes. I think I got that right, correct?
Pyosins, everything else right.
Oh, pyosins, okay. He started out getting his BS at the University of Connecticut, where he graduated summa cum laude. From there, he went on to get his MS at Yale University in the Molecular, Cellular, and Developmental Biology department, and he completed a PhD in the same department after finishing his master's. He then went on for a postdoctoral fellow at Harvard University, where he worked with Richard Losick. I can remember giving a Losick paper at the University of Zurich in Switzerland on activation of sigma factors upon sporulation. And I remember nobody in the audience really cared about it because it was a medical microbiology laboratory. They wanted clinical stuff, but I did it anyways because I loved the work. I thought it was fantastic. So lucky you. And he's been here at Oklahoma State University from 2017, and now he holds the position of associate professor. Since he's been here, he's won a lot of awards: Outstanding Faculty Mentor Award, OSU Excellent in Advising, Distinguished Teaching Award, Junior Faculty Award for Scholarly Excellence, and Excellence in Research Mentoring Award, all at Oklahoma State University. That's a lot of awards for any faculty member to win in their entire career. Matt seems to have done that fairly quickly. He has 31 publications and reviews, including Nature Communications. That was his most recent publication. Nature, of course, is one of the premier science journals out there, and that's just excellent that he got some publications in there. But he's also published in M-Sphere, Journal of Bacteriology, Molecular Microbiology, Cell, and then Nature Cell Biology, and proceeding to the National Academy of Sciences. So he's really done some outstanding work, which is recognized by the superior nature of journals that he's publishing in. He's given 25 invited seminars, and he's been steadily funded through the NIH and OCAST. It seems to me by reading his CV, he doesn't miss an opportunity to apply for grants. He serves or has served a great deal of departmental and scientific society committees. And from 2018, he's been a review editor for Frontiers in Microbiology and Frontiers in Cellular and Infectious Microbiology. He served on a number of research grant panels, obviously a lot of those being NIH, closely related to his research, but also the Deutsche Forschungsgemeinschaft, which is a German review panel for research. He's also had a huge impact on the graduates and undergraduates that he mentors in his laboratory. And so far, he's trained one postdoc. He's graduated 4 PhD students from Oklahoma State University. That's a fairly large amount in the amount of time he's been here, and has 4 active PhD students right now. He's also completed 9 accelerated master's students, which is fantastic. What I'm so proud of is that he's managed to find time for 47 undergraduates in his laboratory. That's huge. Undergraduates, we adore you, but some of you are the most difficult people on the planet to teach research skills to. And it can be costly, but boy, once you guys get into it and that independence takes off, it's the best thing in the world. Certainly for me, it has been throughout my career. And then many of these undergraduates go on to med school and PhD programs. It's just huge success. And also, he takes the time to make sure all of them present or find their names on publications. And I just want to say congrats to all your success with all your students and your OSU career. And with that, let's get on with your talk.
Thank you, John, for a really nice, generous introduction. And thank you all for taking some time out of your Friday afternoon to hear me talk about some of the research we're doing in our lab. This is primarily the work of Robbie Kodka, who's the postdoc in the lab right now because he just graduated. He's sitting back there. So if you have any really hard questions, you can ask him. Today, I'm going to tell you a story about something with which we can all identify. Stress. I might feel stressed giving a talk. You know what stress is. But not only do humans and animals experience stress, but even our single-celled friends, they also experience stress. Of course, not psychological stress, but it is stress due to their environments or their own internal conditions. And one of the definitions of being alive is to sense and respond to stimuli in your environment, including stressful stimuli. And so what I'll tell you today is about Bacillus subtilis. It's a bacterium that has a rather unusual mechanism for sensing and responding to stress using an alternative sigma factor called sigma B. I'll tell you more about that in a minute. I'm going to tell you how we use microfluidics and coupled to fluorescence microscopy to look at stress responses in single cells over time. I'm going to tell you who is in this thing called the stressosome. Don't worry, I'll introduce you to the stressosome. But the identity of the proteins in there impact the timing and magnitude of the consequent sigma B response. And that the dynamics of that response actually affects the way cells, of how fit they are under stress conditions. Finally, I'm going to tell you some surprising new data. I'm going to talk about how we took a mutagenesis approach to figure out how this system senses stress. And we were surprised with the data that we got, which revised the model that has been standing in the field for some 30 years. So let's get started. So Bacillus, unlike many bacteria that principally use things like 2-component systems, which are proteins that are embedded in the membrane so they can see what's going on outside the cell to modify a genetic program inside the cell thanks to some stimulus. What Bacillus uses is this thing called the stressosome, or rather these things called stressosomes, which are cytoplasmic and in this cartoon look like little gumballs. And they are within the cell and they evidently sense and respond to environmental stress. We need them in order for these bacteria to respond to environmental stress. And the way that they do that, they recognize external stressors. Things like the classic ones are ethanol and salt. Yeah, like the same ethanol that's in tequila and rum. And it's salt, like table salt. They respond to antibiotics and heat as well. And when they experience these stressors, the stressosome gets involved. I'll tell you how in a minute. And the response converges on an alternative sigma factor. And just to remind you, in case you don't work with bacteria, sigma factor is the protein within RNA polymerase that tells it where promoters are. So it binds to the -10 and -35 sites of bacterial promoters and coordinately expresses a whole group of genes that have promoters that are the cognate promoters for sigma B. Sigma B, in fact, so the normal sigma factors are housekeeping sigma factors. They just keep the genes on that cells need to stay alive. Alternative sigma factors only come on in certain conditions, like stress conditions. And in fact, the first alternative sigma factor ever to be discovered was sigma B in 1979 in Rich Lozic's lab where I did my postdoc. So here I am carrying the torch, continuing this tradition. Sigma B, the promoters that respond to sigma B or that correspond to sigma B are stress response genes, some like 300 of them that turn on to get the cells through some stressful condition. And this is just a cartoon, but this is about roughly the timing with which these come on. Cells experience stress, and the sigma B driven genes turn on, and then they turn back off. Right, peaking usually at about 15 to 30 minutes after the stress arrives. And what we're trying to achieve in our lab, just to tell you the overall goal here, we want to know exactly how the system, how these stressosomes work, what they're capable of, and why they're important for cells. We want to know it at every single level, from individual proteins that compose the stressosomes and even individual amino acids that compose those proteins, to the stressosomes themselves, to individual cells, to how they affect the entire population and the fitness of that population. Okay. So how does sigma B work? Alternative sigma factors are at the ready, but they're kept off until they are needed. Right? If you study a eukaryotic system, NF-kappa B works the same way. It's pre-made, and then you just turn it on when you need it. It's just like, I don't know, firemen. When there's a fire, you don't want to train firemen. You have firemen ready, and they just are a phone call away. And so sigma B normally, when cells are not stressed, is kept in an off state by a thing called an anti-sigma factor, whose one job in the world, well, almost one job in the world, whose main job is to bind to that sigma factor and keep it off. So we call it an anti-sigma factor, all right? We name these alphabetically. This is W. All these proteins are actually RSBW, response mediated by sigma BW, but I'm just going to call them by their last letter, yeah, because we're on that kind of basis. So W binds to sigma V, to sigma B, keeping it off, okay, until there is stress. When there's stress, what happens is this protein here, V, which is normally phosphorylated, gets dephosphorylated by a phosphatase. I'll tell you about that phosphatase in just a second. And when V is dephosphorylated, W would rather be with dephospho V than with sigma B. So there's a partner switching mechanism that occurs. So W switches from sigma B to V, and now that leaves sigma B free to interact with RNA polymerase and transcribe all those stress responsive genes. Okay. So far, so good? So whether sigma B is active or not is controlled by the phosphorylation state of V. So we need to have some kind of a phosphatase for V. By the way, the system tries to turn itself back off. I said almost its one job in the world. Its other job in the world is as a kinase for V. So W ends up rephosphorylating V so that it can go stick back to sigma B. So the system turns itself off. It's not going to be on forever. But to turn it on, we need some kind of a V phosphatase. Okay, so what's the V phosphatase? All right, it's U. That's U is a phosphatase for V. But okay, but if U is always around and dephosphorylating V, we'd always have this stress response on. So something has to control the activity of U, whether it's a phosphatase on or off. Guess who does it? T. Yeah, we're just stepping back through the alphabet, as you can see. The activator is RSBT. And so the access of RSBT to U, or the availability of RSBT, is critical to turn this system on. Okay, so then, it's just like one thing after another. So what makes T available or not? That's finally where the stressosome is. So the stressosome is made of two different types of proteins. R, turns out there are multiple R's, that's why I have different colors. I'll talk more about those in a minute. And S, which is a scaffolding protein. We have 60 proteins here. We have 20 S and 40 R's. And they make this big old complex. When the T is bound to it, it's like almost 2 megadaltons, so big. And there are many, like 10 to 20 of them in every cell. And so to this complex binds T, the light gray ones, and it just sequesters T, so T is not available to activate U, dephosphorylate V, cause the partner switch, activate sigma B. Okay, so this stressosome, somehow it senses stress, and when there is stress, it lets go of T. Right, and there are some things that I'm not even depicting here in this particular diagram. T gets let go of, but T, it turns out, is also a kinase, and when there is stress, T phosphorylates R and S at the same time as it gets let go. All right, I'm oversimplifying just a little bit, but that's the important thing to know. It's also kinase, because I'll turn to that way later in the talk, so that's your retention. And when T is released, then it can do all those steps that lead to the activation of sigma. And if you read reviews about this, like there are some fundamental questions that still remain unanswered. Like we don't know what this thing senses or what the sensor is. If you read the literature, they'll say, okay, it's sensed by some unknown mechanism, and that's communicated to T's kinase activity by an unknown mechanism, and then T phosphorylates RNS and gets let go. Okay. So who is the sensor? We really don't know, but for a long time it's been presumed to be the RSBRs, the colorful ones here, and that's for a couple circumstantial reasons. What you're looking at here is a cryo-EM structure of the stressosome, and so the N-terminal domains of these RSBR proteins are the ones shown in yellow here. They're extending out like turrets from the core of the stressosome, so they're in the right place to be sensors. They're out waving in the cytoplasm. Okay, and then by analogy, there's an RSBR-like protein that has a very similar C-terminal domain that sticks into the core of the stressosome. But its N-terminal domain, instead of being the non-heme globin dimer that is normally RSBR, instead it has a LOV domain, and it senses blue light. So there's another protein that's similar, and its domain that's in this position, we know what it senses, and it senses blue light. So by analogy to that, we say, okay, maybe these are also sensing something because they're in the same place, same position on a homologous protein. Okay. The other thing is that there are 4 different types of RSBR. There are 4 different paralogs encoded in different places in the genome. That's why I'm showing 4 different colors. And so these paralogs have very similar C-terminal sequences. They're the ones that are on the inside. But their N-terminal sequences differ quite a lot. And their C-terminal sequences are very similar to RSBS, which is the core scaffold inside. Yeah? And you can see here, these are the T and the S. That's just the serine and threonine residues that get phosphorylated by RSBT when they let go of RSBT. So a long-standing question is like, why have these variants? We know that bacteria don't keep genes around that they don't need, so they must be doing something. And there have been some attractive ideas about why have these different ones. Maybe it's because it senses a lot of stuff. It's the general stress response. You can sense ethanol and you can sense salt. So maybe you need a lot of sensors, and these are sensors. And you need a lot of different sensors to sense different stuff. Although there's not very good evidence for that. In fact, all the evidence that we've gotten counters that hypothesis. But in any case, we wanted to analyze these and try to see if we can distinguish one from another, see if they're functionally distinct. So I've got to tell you a little bit about how we analyze these in our lab. So the classic way to analyze something that's really small, like bacteria, is you do some kind of a assay in bulk. Yeah, you take a lot of bacteria at the same time, and you see what they do. When you do a Western blot, you're doing a bulk assay. When you're doing an OD, you're doing a bulk assay. When you're using some kind of a reporter, you're doing a bulk assay. And so if we imagine just like a cartoon here, where we're looking at greenness over time, and as time goes on, the population gets more green, maybe we think, well, what's happening is at an early time point, all of the cells are light green, and at the late time point, we have a lot more greenness. They're a lot greener. But consider that this pattern gives you the same bulk pattern. Yeah, rather than having lots of cells light green, we just have a few cells dark green, and then over time we just have more cells that are green. These 2 dissimilar patterns at the single cell level give us an identical average. So we have to kind of try to come up with methods that let us look both at average and at what's happening at the single cell level. And there's a trade-off there. The bulk culture methods are nice for these kinds of experiments that we do, because when we want to stress cells, it's easy to grow cells in a flask, and we want to stress them with ethanol, for example. We just squirt in some ethanol, and now suddenly they're stressed. So we have some environmental control there. But we don't get any single cell data when we're looking at something in a flask, because we're looking at millions of cells at the same time in any assay we do. Okay, but you could do microscopy. The classical way to do microscopy in single cells, live cell microscopy, is you put your cells on an agarose pad, and you put glass cover slip on top, so the cells are squished there and immobilized, but they're not hurt. It's like I stood up against a plate glass window and someone pressed a mattress behind me. I couldn't move, but I'm not being hurt. That's what we do to cells. But that's really hard to change in terms of its environment. So we can't squirt anything in there. And in any case, in both methods, whether we have a flask or we have what's essentially a tiny flask on a microscope, As the cells are growing and dividing, they're using up nutrients, they're secreting waste products, they're changing their own environment in ways that we don't know or understand. It's continuously changing. So that's not good for the kind of observation we want to do when we want to look at the response of cells to some stressor, right? And because they're these closed systems in a flask or in this tiny flask, we can only watch them for so long before they run out of nutrients and they just stop growing. All right, so to try to get around, circumvent some of these problems, we turn to microfluidic devices, which allow us to look at something on a microscope, but in an open system where we can alter the environment in any way that we choose. And we use a system that's called the Mother Machine. I'll tell you why in just a minute. We manufacture our own devices in-house. You can see the scale bar here. So these guys are actually really small. And on this device, which is made of a flexible polymer called PDMS, we bind this covalently to a glass cover slip. And if you were to zoom in here on each of these channels, you would see what looks like teeth on a comb. And if we further zoom in on there, what we actually have is little channels that are just the right size for bacillus cells to fit in single file. And they've got a little shallow channel around them, so they get plenty of medium exchange so that they're in a totally uniform environment as we flow medium by. And these channels have an end to them, so as cells grow and divide, they push their progeny up and out into the medium flow. And the one cell that sticks around that we can watch is the one that is at the bottom. Yeah, we call that one the mother cell. And so this type of microfluidic device is known colloquially as the Mother Machine. Yeah, which sounds like vaguely badass. Okay, so we watch those mother cells and we can see what happens over time. I don't want to be better than anybody. Like, it sounds fancy. This is what it actually looks like, right? Generation 1 used a pipette tip box as our for switching medium from non-stress to stress medium with our fancy binder clip pinch valves there. And this is like generation 2.0. Actually, I 3D printed over in the library, so you don't have to use this nasty old pipette tip box. It's epoxied in place. All right, but this is on an inverted fluorescence microscope, and all the plumbing's on the backside. That's why you have to have an inverted fluorescence microscope. So we're watching everything happen from the bottom of this where the glass is. Okay, so when we do this, when we look at cells, we can have something like a constitutive fluorophore, in this case RFP, so we can see where the cells are using fluorescence. But we can also just use phase contrast microscopy. That works fine too. And then we have a GFP reporter that's driven by a sigma B responsive promoter, so that when sigma B is on, when the cells are responding to stress, they turn green and we can see it. All right, so we just watch over time. What a chimograph is if I take a picture at time point 1, and then a picture at time point 2, and a picture at time point 3, and I just line them up in time, you can see that it's dark, and then we add stressor, and they get bright, and then they turn dark again. And if we take these values and we just take averages so that we can plot either a single cell, or in this case, it's a whole ensemble average of cells. We see that they're off, they're off, they're off. We add stress, they all turn on, and they turn back off. So we get the real data that look a lot like that cartoon that I showed you before. Make sense? This is what the data look like. Okay. So here's an example of cells responding to ethanol stress, just so you can see what this looks like. At some point, we're going to add ethanol stress. You're going to be looking at these cells at the bottom, the mother cells, and we're going to see what they do. We add some stress. Woo! They all get bright at the same time. Then they get dim again. Yeah, that's it. And they don't really get bright again, as you can see, even though we can watch them for hours and hours and hours, a lot longer than we could ever do an experiment in a flask. So when you do an experiment just like this and we plot the data, it looks like this. We have all these individual cells. They all turn on, turn off at the same time. It's a synchronous response. It's a transient response. And we plot the average. It looks like this. I left this in here with these asterisks. What are these? Are these other responses? No, they're clogs that are happening in the microfluidic device. These experiments are extremely prone to failure. They're really hard to do. And I don't want to minimize the amount of time it took former graduate student Chris Ham to do a lot of these experiments. He did the ones that you're looking at right now. So these are what wild type cells look like. Transient, synchronous response across cells. They're all behaving in a stereotyped way. Okay, but what happens if we take cells, so those are wild type cells that have all of the RSBR paralogs in. But now we can use the facile genetics of Bacillus subtilis to engineer strains that only make one or another of the RSBR paralogs and ask how they differ. Yeah, so I'm just going to give you, just for time's sake, I'm not going to show you all of them. We've done them all and more, but I'm just going to show you one. All right, and we can ask questions like, do they respond to one stressor and not another? It's like, do they actually act as different stress sensors? The answer to that question is really no. And do they distinguish among stressors? Do they change according to stressor? And the answer to that question is overall yes. Okay, so now we can see here what some of these look like. This is all Chris's work. And so he's taken wild type cells and he's exposed them to different stressors, not just ethanol like I showed you, but also peroxide and salt and acid. And you can see that every time you get responses with different magnitudes, but they always look the same and with respect to being transient and they're synchronous. All the cells are behaving in a stereotyped way. When we take cells that only have this one RSBR paralog RSBR-A, they look like the wild type. They're also transient and synchronous, which by the way suggests that A dominates the wild type response. But when we look at another one, here's an example of ones that only have RSBR-D. And in this case, look how different the result is. It's not transient. It's sustained on an average basis. And what's happening in individual cells is they're all turning on and off and on and off and on and off, but asynchronously. So at any given time, some of them are on and some of them are off, which gives, as an ensemble average, what looks like a sustained response. Yeah, we see that for ethanol, we see that for salt, but we don't see it as much for pH or for peroxide. In that case, it behaves more like the wild type. It's more transient. So yes, they can distinguish among different stressors, but all of them respond basically to all stressors. So that kills the hypothesis that different ones specialize in different stressors. Okay. So then we wanted to ask, we see these different response profiles, and remember, I just showed you 2 examples because otherwise we'd be here all day. We tested like every single one of them, okay? And we take 2 that are very different. For example, RA only cells with their transient responses here to ethanol and salt, and these RSBR-D only cells that have these sustained or repeated responses to ethanol or salt. And we asked, do they have different fitness? Does having a different shaped response change how fit these cells are when we are exposing them to some stressor? And the way we did this is just really simple conceptually, although these experiments are surprisingly hard to execute correctly. But an army of undergraduates did this over a period of years. And again, the concept is simple. We just take one of these strains, RSBR-A only, for example, and RSBR-D only, and we mix them together at a 1-to-1 ratio, or as close as we can get to a 1-to-1 ratio, and we grow them in the presence of stressor, keeping them in exponential phase, so it's like being in the mother machine, keeping them in exponential phase for 9 hours, and then we plate them out, and we take pictures of them, and they're differentially labeled so that we can tell one from another, and we see whether the proportions change. Got it? All right, and it's pretty cool and fun experiment to do. We actually invented a way to do it ourselves. We take this, police grade flashlight that's super, super, super bright. It's like $100 flashlight, which is the most expensive flashlight I've ever seen, but like one of the most cheap pieces of lab equipment I've ever seen. And we take that flashlight, and it's got a 1-inch aperture at the front, and we just use electrical tape to put a Thorlabs bandpass filter over it, and suddenly we have a fluorescence excitation illuminator that can give us either shine blue light for GFP fluorescence or yellow light for RFP fluorescence. Then we just take a piece of long pass glass that only lets wavelengths beyond a particular wavelength pass through. We just hold it over our smartphone camera, and we take a picture of the same plate. This is the same plate. Look, all the ones that are green are not red, and all the ones that are red are not green. So all I have to do is count and see whether the proportions change. You with me how this works? Give me a vigorous nod so I can see. Yeah, okay, thanks. Okay, so that's all we do. We count these, and then we can use that to make these competitive index graphs. And so we can see, is one dominating over the other one? And if they're in a dead heat, it stays right around 1. This gray bar is our experimental error. But if one's winning and another one's losing, we see it either above or below here. And you can see highlighted here, I'm just talking about A and D today, just for the sake of time. But you can see when we take RA versus RD, A wins consistently in ethanol. A beats D. But guess what? When we put them in salt, it's totally different. Now D beats A. In fact, D beats pretty much everybody. It's really interesting. It outcompetes all comers. And so our model is that the sigma V response really does influence fitness. That a transient response is advantageous in ethanol, but it comes at a cost in salt. But this type of response is advantageous in salt, but then there's a price to be paid in ethanol. And so we wanted to test this hypothesis a little bit more rigorously. And so we made a prediction that if we get rid of RSBRA in cells, they'll lose their transient phenotype, their transient response, and then they'll behave more like RSBRD only cells. And thus we ought to see both a shift in their response profile from an A-like more towards a D-like, and we also expect to see a fitness shift. So from being better in ethanol to being worse in ethanol, better in salt. So we did this experiment. We knock out RSBRA from wild type cells, and you can see in ethanol, we broaden the peak. So it's the longer lived response now, especially in salt. We have like the real sustained response when we get rid of RSBRA. And we look at fitness, we see that now we're comparing the A mutant versus the wild type. And by getting rid of A, indeed, we now have a fitness deficit in ethanol. And it's not statistically significant, but it trends toward a fitness benefit in sodium chloride. So at least we can say that having this more extended response it's more of a detriment in ethanol than it is a benefit in sodium chloride. But this supports our hypothesis that the shape of the response matters for fitness. And indeed, when we knock out sigma B entirely, we don't have any significant effects, again, suggesting that it is the sigma B response that's important for the change in fitness, because when we get rid of sigma B, so does the change in fitness diminish. Right? It doesn't diminish to zero, but it largely diminishes. Okay, you with me? Cool. All right, so let me just summarize where we are. Just bring us back before I tell you about all this interesting, surprising stuff we found. So indeed, the identity of these paralogs in the stressosome influences the profile. Who's there changes the shape of the response. When we have wild type cells, they look the same as RA-only cells with a transient synchronous response, whereas the other ones show different, usually more extended or repeated responses, which depend on which stressor is there and which one is present, which RSBR paralog is present, that is what the response looks like, changes fitness in different stressors. Okay, moving on. I told you already, critical events have to be release of RSBT from the stressosome. RSBT, as long as it's stuck to the stressosome, can't activate sigma B, but when it gets released from the stressosome, then it activates sigma B. Fine. And I also just want to remind you why we thought that RSBR is the sensor. We didn't know, we don't know what's being sensed there. And again, we think it's the sensor because it has these domains that are sticking out into the cytoplasm in the same place where the blue light sensor has its blue light sensor, and because it's just in the right place. And we want to know, if it's a sensor, what residues in there are acting as the sensing residues. We figured that the easiest approach to try to find residues that are involved in sensing would be to try to find mutants that could no longer sense stress. We're going to mutate one residue at a time to alanine, just as a methyl group as its side chain, and see if we can find something that no longer can sense stress. So at least suggest to us what part of this protein might be the sensor. Okay, so to do this, it was a little bit complicated. Robbie had to do a lot of work to do this, and this is all Robbie's work now. And so I haven't even told you that there's a whole other branch of stress response, a response to energy and starvation, right? And so it converges on the path that I showed you here. Instead of using U as the phosphatase for V, it uses this protein P as a phosphatase for V. And so sigma B can also be activated by starvation. So we have to get rid of that. So we have to get rid of lots of stuff. We have to get rid of P, so we isolate only the environmental stress response. And then we're going to get rid of YTVA, which is the blue light sensor. And then we're going to get rid of all the other RSBRs. But before we get rid of the last RSBR, the prediction is if we get rid of the whole stressosome, now T is going to be on the loose all the time, right? And so we should have a constitutive stress response. But we don't want that because that will make cells sick, we think. So before we get rid of the whole stressosome, we're first going to disable the environmental stress response by getting rid of U. Okay, and now by getting rid of U, now we can safely get rid of A. And now we can have a system where we can test mutants by adding back to this system RSBU to reconstitute the system. And at the same time, we add in a mutant version of RSBRA with one single amino acid mutated to alanine. You with me? That's how this works. And the cool thing about it, so here's the region that we scanned. Every one of these, we changed, just about every one, we changed to alanine if it's not already alanine. Here's just a picture of what I just told you. So we put back into our cells this little construct that's got our mutant RSBRA and RSBU to reconstitute the system and test our mutant. And then, of course, we have to have a reporter that tells us whether these cells can respond to sigma B, or excuse me, respond to stress or not. And the cool thing about the screen is that this is what wild type looks like. We add stress, this is a beta galactosidase assay, so just looking at that reporter, that LAC-Z reporter. We add stress, we see a response. That's exactly what we expect for wild type RSBRA. This is the actual data for when we add back a wild type with RSBU. So we know the system works. Now, if we find a mutant that's sensing dead, we expect to see nothing. We add stressor, and nothing happens. However, if we find a mutant that somehow screws up the protein so it can't fold right, and it can't sequester T in the stressosome, then what we should see is something like a constitutive response. So we thought this was really clever because we can isolate phenotypically only ones that are sensing dead. Yeah, so this is Robbie made all these different constructs and started testing them. It's a huge amount of work. Huge. I mean, he's made like hundreds and hundreds of strains and done so many of these assays. Each one takes a long time. And what we found is we didn't find any sensing dead mutants. How about that? Ooh, and no constitutively active mutants. We found some mutants, a very small minority, that have a stronger response than wild type. We found a huge number of mutants that have a weaker response than wild type, but none of them are zero. Yeah, and we had reviewers ask us, what are these like really low ones? Aren't they like pretty much zero, or are these actually sensing dead? And so we take those mutations and we give it like a little more stressor and they go up. So it's a real response. Okay, so there are a lot that lower it, and then there are some that don't really change it much at all. But you can see that all these different ones have different shapes too. Like we're basing high, low, or not changed based on where the peak is relative to the wild type, whether it's within 10% or not. But look at the different shapes you get too. Some are earlier than wild type, some are later than wild type, some last longer than wild type. It's pretty cool. All right, so what this told us is, well, let me show you one more slide in a minute. All right, so this is just a little icon showing that like most of these in red are the ones that lower the response. Yeah, the one that's in green raises the response. We wanted to test some of these a little bit more rigorously. So instead of using the strategy I just described to you, where we take everything out and then we put back in U and RSBRA, we can actually do in this species, we can just replace right at the native locus the regular RSBRA with a mutant version of RSBRA. We call this allelic replacement. And we tested some of those using allelic replacement, and we see that these ones have an even milder phenotype, although the same pattern emerges. We have some that are higher, some that are lower, some with altered timing, some that don't change it much, although they change the timing, right? It comes up, comes down, we get a little secondary response with this one. In one respect, and this is just showing you with the ones that we tested, and the ones with asterisks below are ones where it was different in our two methods. Yeah, we trust, if there's a discrepancy, we trust this one more because it's less perturbative. We're making a more subtle change to the genetics. So in one sense, this was disappointing. We didn't find the sensing dead mutants we set out to find. But in another sense, this is really exciting because what we have here, what we have demonstrated rigorously with all these mutants, is that this is a system that's easy to tune and hard to break. When we make mutations to it, it doesn't kill the way that it responds to stress. It just changes it, it just tunes it. So it's really evolutionarily robust. This is like a molecular machine, and when you screw with the machine, it doesn't break it, it just changes the way it works. It's sort of like a gearbox. You give it the same input every time, and the output changes somewhat. So cool. Okay, and like we find that many of the mutations that have the largest impact are at the edge of these alpha helices here. And so it could be that these are hot spots that either influence T being let go, work still has yet to be done, or that somehow influence the sensation of stress. We also want to do, we have very little bit of data here, just to try to back up the idea that these are evolutionarily robust proteins, that they're robust to mutations, right? I'm not trying to prove anything here. All I'm doing is suggesting. So we took 50 random sequences from NCBI Bacillus subtilis, and we just looked at the frequency of mutations that occur in the N terminus of RSBRA versus the C terminus of RSBRA, which is the more conserved region. And we would predict that this is robust to mutation, as we found, and we see more mutations in this section than we see here. Yeah, so that at least we have some evidence backing up the idea that this is a tunable system that's robust to mutations over evolutionary time. But in any case, the fact that we didn't find any sensing dead mutants just made us do a rethink. And to ask, all these years we've been thinking that RSBRA is the sensor. Is it really the sensor? And so we got rid of all the RSBRs. I told you before that the prediction there is that we're going to have a constitutive response, because if we get rid of all our RSBRs, we shouldn't have a stressosome. We have nothing that can hold on to T. T should always be on the loose, activating U, dephosphorylating B, causing the partner switch and activating sigma B. And to our great surprise, when we knocked out all the RSBR paralogs, indeed, we start higher. Even without stress, there's some response, but there's a clear stress-inducible response that's stronger and longer lived than in the wild type. So there's still stress-sensing activity here. Robbie got this data, and I'm like, I don't believe it. Like, this can't be right. Do it again. Did it again. It looked exactly the same. Okay, I've already told you about 5 RSBR paralogs, A, B, C, D, and a blue light sensor YTVA. Would you believe actually there's a 6th? A 6th that's been abandoned by the Bacillus subtilis. It is split by a frame shift mutation. So we figured it doesn't do anything. And so we knocked it out just to be safe. We thought maybe this one's actually active. But, you know, this isn't the surprise. It's not active. We knocked this one out. It looks exactly the same. Right? But that was strong data for me, too, to believe it. This is now a different strain with another deletion relative to this one. So it already has a ton of deletions. And look how closely they resemble one another. They start high. They even have this little dip. And they have this strong, long-lived response to stress. Still stress responsive. Okay, so we get rid of all the RSBRs. What's left? There's RSBS. That's scaffolding protein. We thought, gosh, could it be possible that RSBS is doing the heavy lifting? That RSBRs is picking up the slack? We get rid of all RSBRs, and we still have this stupid scaffold that we thought couldn't do anything, but it actually is doing something. So then our strategy was, let's just knock out RSBS. If we just knock out RSBS from wild type cells, we're missing the scaffold. Also predicted that we're not going to have stressosomes because we're missing the scaffolding protein. Let's see what happens then. So we knock that out. It looks the same. All right, we get rid of the scaffold. We shouldn't have any stressosomes. Look, it even has that little dip again. Strong, long-lived response. This is all pointing towards we don't need the stressosome. We can still get a stress-inducible response even without the stressosome. And so far, I've showed you data where we got rid of all the RSBRs, and I showed you data separately where we got rid of RSBS. So now, what happens if we get rid of both? Okay, it's a little more complicated than it seems because all these genes are in an operon together that I'm going to draw for you because it's going to come back later. I'm a professor, so I just keep dry erase markers in my bag. And so we have all these genes in one operon. We have RSBRA, and then we have S, and then we have T, and then we have U, and then we have V, then we have W, then we have sigma B, then we have this protein or this gene, RSBX, and there's a promoter up here, which is a sigma A driven promoter, which is just a housekeeping sigma factor. And then there's another promoter here that's a sigma B driven factor. So sigma B feeds back on its own operon, okay, and feeds back on itself. And in fact, the reporter that we're using here is a reporter that's hooked up to the sigma B, the RSBB promoter. Yeah, it's this promoter that we actually use to read out for sigma B. All right, but because these are all in an operon, if we're going to get rid of R and S, we were afraid we were going to screw things up with T. So instead of doing just deletions of these, instead we put early stop codons in those genes to block them from being translated. So we won't get the proteins, even though we haven't messed up the genes. Yeah, you with me? Okay, so that's what we did next. So we got rid of both using these early stop codons, and whether we get rid of everything except RSBRA, stopping S with a stop codon, or if we get rid of everything with stop codons, it looks the same. We still see it's a little higher than before we had the stress, it's higher, doesn't stay off as well, but we still see a stress-inducible response. Okay, so we've now ruled out any component of the stressosome except for T. T is the only thing that's left. So, do we need T? Yes. We take that same strain that's lacking the stressosome, and we get rid of T, and it kills the response. Nothing. All right, so that's a deletion of T. But if we put an early stop codon on T, looks the same. Nothing. T is what's needed. Could T be the sensor? When we put T back, ignore this little one for a minute, I'll talk about that later. When we put T back, we can restore a response, but it's not a very good stress response. This is where we add stress to the dotted line. See, it's not a very good response there, but we at least restore activity, telling us that T is the linchpin here, right? And I'll talk in a few minutes about why this doesn't have much of a response, or why I think it doesn't have much of a response. So if we put T back on a plasmid, we can at least restore sigma B activity, if not an inducible stress response. Okay. Well, I'll get here more in a minute, but you can see that T also has kinase activity, right? Kinase activity towards the stressosome, R and S. These are cells that don't have any stressosome, so we would figure that the kinase activity is totally irrelevant, but it is not. We get rid of kinase activity with this well-characterized substitution, we kill activity, or at least we decrease it by a lot. Okay, so that's still a mystery of why, and I'll return to that mystery of why kinase activity is still important. So we looked at these in single cells. We looked at the stress responses in single cells using the mother machine. First of all, we see, yeah, we definitely get a response. And what it looks like in single cells is a repeated response. They turn on and off and on and off and on and off and on and off. A lot like what we see in those RSBRD only cells that I showed you before. And what this tells us, like the fact that we see this is two different strains, right? This one's missing all of the stressosome components, and this one's missing just the RSBRs. It still has RSBS, but you can see that they look the same. A couple of things come out to us we can learn from this. All right, one of the things we can learn is that this pulsatile activity we see, this on and off, back when we saw that same response in cells that only had RSBRD, we might be tempted to think, oh, like what's happening is T is coming off the stressosome, and then it's getting recaptured by the stressosome, and then it's coming off the stressosome, and it's getting recaptured by the stressosome. We know that that's not happening because we're got rid of the stressosome. And we still see that pulsatile activity. So that means that pulsatile activity is not caused by recapture release dynamics. The second thing we saw is, look, it's a graded response. The more ethanol we add, the stronger the response gets. And so there's this paper that came out something like 15 years ago that suggests, so you don't have to read all this stuff, but all this is a feedback loop that was a really nice way to explain the way that pulsing works. That, like, you get, it's a paper with this system suggesting that you can get pulsatile activity, and specifically suggesting that when you give cells more stress, they pulse more frequently. All right, it's a beautiful model, and it was very surprising. It was published in Science magazine because the idea was, instead of modulating the amplitude of the response, it modulates the frequency of the response. And indeed, if you had a higher frequency response with more ethanol, you would see a higher average, because on average more cells are on because they're more frequently pulsing. We have the ideal system to test that hypothesis, and what we saw was the opposite. So we look at how big the peaks are, what the amplitude is, and with more ethanol you can clearly see that the peak height goes up. And if their peaks were getting more frequent, we would see the frequency go down, but we don't see the frequency go down. We see it go even up a little bit. So these are not frequency modulated, they're amplitude modulated. In fact, we have never been able to recapitulate that result showing frequency modulation. So I'm believing it less and less every day. Okay, I'm going to wrap up in just a minute. First, we have a revised model for how the stressosome works. We don't have data that the RSBRs and RSBS, the stressosome itself, are really sensors that like tell RSBT what to do. We think instead they're like advisors. Do they have an important role to play? Yes, they do. Clearly, they keep the response off in the absence of stress, and they affect the timing of the response. They turn it off after it turns on, so you get a nice sharp response. But they're not strictly needed to get a response. Yeah, this is just what I said. Right? When the stressosome is there, we get a more rapid response, we get a shorter live response, clean signal, good signal to noise ratio. And so we think of it as like the stressosome is like this, as I said before, like a gearbox. It can modulate the output to different inputs. That's exactly what a gearbox does in your car, right? In different gears, the engine spinning at the same speed produces the wheels spinning at different speeds. Because we showed that pulsatile sigma B is not due to the stressosome, it's downstream of the stressosome. And we think that RSBT may be the stress sensor. In fact, like even in the stressosome, When there's stress, it's RSPT that turns on its own kinase activity, phosphorylates RNS, and gets let go from the stressosome. We don't know where that stress sensing activity resides in T yet. We're working on. All right, so I'm just tell you where we're going from here and give you a couple teasers. All right, one student in the lab is working on these point mutants that Robbie made that change the response profile and is asking, do they impact fitness? When we lower the response or we raise the response or alter the timing of the response just with single amino acid substitutions, does that impact fitness? We're looking at those altered response profiles at the single cell level. Do they pulse? Do they not? Do they have a single transient response, but it's longer lived? We have some intriguing data that's that we do still have synchronous responses, but that they last longer. Right? We want to know what RSPT senses. It responds to all these different stressors. What is it actually sensing inside cells? We don't know yet. How does it do it? What parts of it are important? Well, we know one part that's important so far, and that is its kinase activity. I showed you that already. We get rid of its kinase activity, even though in theory we have nothing left for it to phosphorylate, and it still doesn't show a good stress response. So that kinase activity is somehow important. And the system lets us now revisit a lot of old assumptions, except in the absence of a stressosome. And I just want to show you two of those. Right? This is what I showed you before, that the kinase activity is important. We get rid of the kinase activity and we can't reconstitute the sigma B response. But there's also something I didn't show you before, and that is that there's a phosphatase, X, it's this one over here, X, that undoes this phosphorylation so that the stressosome can recapture T. Yeah? In the absence of a stressosome, we would say, so like when there is a stressosome, let's start there. When there is a stressosome, if you get rid of X, you get a much longer lived response because the stressosome can't recapture T in a timely way. Make sense? If we get rid of the stressosome, X shouldn't have a substrate, so X should be totally irrelevant without a stressosome. Okay, guess what? It's not. So there's in just a stressosome deleted cell. And if we get rid of X2, we get an even greater response. So not only is the kinase activity important. This data actually I haven't shown you before. It's the same strain here, except now T has that one mutation that stops its kinase activity, and look, it kills the response. So we need the kinase activity, and we also need the corresponding phosphatase activity, even in the absence of a stressosome. So there are obviously more substrates for both the kinase and the phosphatase that were unappreciated before. Pretty cool. Okay, we don't know what the target is yet. All right, I got one other thing to tell you. I'm going to skip this. And that is when we take cells and we overdose them with RSBT, oops, we give them RSBT from an inducible plasmid and we look what happens. And this is just the data that are plotted here. We don't see this pulsatile activity anymore. Instead, what we see is when we induce T, our reporter comes on, now the mother cell's up here, and it just stays on. No pulsing. And so my favorite hypothesis, and I'll finish with this, my favorite hypothesis is that we're actually getting unanticipated feedback, transcriptional feedback, somewhere up here. Before, we thought that sigma B only fed back to here, and I think it feeds back even farther. And we actually have some data from Robbie that suggests that this is the case. He did earlier in this paper a qPCR to look at expression of T itself, expression of T before and after stress. And you see in wild type cells, you get a boost in expression 15 minutes after you add stressor. Even without a stressosome, you still get that boost. Yeah? If you delete RA and S, where presumably there might be a promoter for T, you lose it. Ha-ha! So that is a hypothesis that we're really eager to test in the very near future. Okay, so that's all I've got for you today. Again, a shout out to Robbie, who did most of this work. Where is he? He's in here somewhere. Do I not have? No, there he is. There's Robbie. And this also showed you work today from Chris, who's now a postdoc at UAB.
There's Robbie.
And he's right back there too. Yeah, I'm not gonna. I'll shine the laser at you. And Niloy has also become a big part of this project, trying to find out exactly what T is doing. And I'd be happy to take any questions that you have.
Great talk. Thank you so much for this. And
Sure.
really good bacterial genetics and whatnot. How many promoters, how many SIG-B promoters are there on the chromosome?
Oh, I mean, there are something like 300 genes that are under SIG-B control. So I don't know if that means that there are certainly fewer than 300 promoters because some of them are in operon.
Yeah, gotcha.
Yeah.
Yeah, but I started, most of your work demonstrates with a single promoter, you're working with a single promoter, correct? Do you see that it's possible that maybe other SIG-B promoters might act differently?
So in the paper, I just didn't have time to show you. We used another separate sigma B inducible promoter, and the shape of the response is slightly different, but it's qualitatively the same.
But you wonder the whole stress response, see modification through multiple promoters.
For sure.
Acting on all of these factors individually, differently, over time. Anyways, that's.
Yeah, no, I think if you were to do a comprehensive study and put a reporter on like every single one, you'd see general patterns that were similar, but you would see specific patterns that differed from promoter to promoter for the exact reason you suggest, that many of them are further modified by other transcription factors, for example.
Yeah, so suggesting that the stressosome is important, but maybe not so important for what this particular.
So in that case, so let me restate the question, make sure I have it right.
Yeah.
So does the stressosome impact different promoters differently?
Yeah.
Like the stressosome itself. But everything converges on sigma B. Like it's sigma B that's responsible for everything. The stressosome doesn't interact with DNA. So I don't think the presence or the absence of the stressosome affects anything except the dynamics of the availability of sigma B. And then anything happening downstream of that is just because of sigma B being able to bind to RNA polymers.
What about the sporulation?
Yeah, what about sporulation?
I mean, these are still appendix.
Yeah, sure does. Yeah, so sigma B is actually not very important for sporulation. There's a different set of sigma factors starting with sigma F that's important for that. And so in sporulation, there's a very well characterized, sorry to get in the weeds a little bit. It was a big topic of my postdoc lab. So as you're getting sporulation, you have the endospore, it develops inside the cell. So there's our bacillus cell. And here's the spore that's developing. And so this is the mother cell, and this is the spore. And what happens is you have sigma F get activated in the mother cell. And one of the things that sigma F activates in the spore, we call it, is sigma G. And then sigma G gets activated in the spore, which then causes activation of, I think it's sigma K in the mother cell, which then causes another sigma factor to get turned on in the spore. And so it's a very very well described at this point, a very specific program of genes that comes on at each time to progressively build the spore and like cause the mother cell to die and give up its life for the spore.
It's cell differentiation is what
that's totally what it is. Students, don't be afraid. Oh yeah, I asked you.
If do you know how cell pigment is affected? Is it the same operon that I don't know, or maybe something else?
Like, so you're asking what it is about sigma B that affects fitness? Well, that's a tricky question. It's experimentally accessible, but only annoyingly. So like what you would have to do, I guess, is you'd have to take different operons and then alter their promoters so they're not as responsive to sigma B, and then see if that had something to do with fitness. Like, so do you see a fitness deficit because we're expressing such and such an operon? So we have to swap that operon or delete the promoter so that you do not now activate that with sigma B. Is that your question?
To identify how it is.
Well, I mean, the fitness as we measure it is simply speed of growth. And so, yeah, I don't know. Nothing comes to mind that would really tell you what's changing the speed of growth under those conditions, unless you could isolate mutations that suppressed it, or you could do you could do an omics experiment where you looked at transcriptomics under sigma B expression to see what was turned on. But like, I have a feeling you'd still be facing a needle in a haystack sort of problem. Yeah. Sorry, I don't have a more satisfying answer.
Which made me think of another question. So in all your knockouts, was there any alteration in growth of the bugs?
No, so minimal. So you get a little slowdown in growth when you express sigma B, like when sigma B is active, but like just by making all those knockouts, no,
Yeah.
sigma B, in fact, in lab conditions is entirely dispensable. You can get rid of sigma B, and whether you have it or not, until you face harsh conditions, really doesn't have any fitness effect at all. Yeah.
So could you restate how you measured the stress response in the cells? Did you use some kind of fluorescent?
Oh.
dye or-
So no, so there are two different ways. If it was in the microfluidic device, then what we're analyzing is simply the magnitude of GFP fluorescence, because we have, it's not really GFP, it's neon green. We have a fluorescent protein that's being driven by the RSPV promoter. In all the bulk assays we use, we're doing beta galactosidase assays, and we're doing them, we're right, okay? Which, so the way we do it in a lab, which is annoying for my students, but we think it's a little bit better. Instead of just doing an endpoint analysis, so if anybody's ever done a beta galactosidase assay, we put this indicator called ONPG, and if you have the beta galactosidase enzyme that's been produced under the control of your reporter, it turns ONPG yellow, and you see how yellow it is. That's sort of the classic Miller assay, is you break open your cells, so you get all the beta galactosidase out, you put the ONPG in, and you let the reaction run for some period of time, 20 minutes or something, and then you stop it and you see how yellow it got. The problem with that method usually works fine, but like when you give it enough time, slower ones catch up to faster ones, so you don't see the difference as well. And so what we do is we actually do a kinetic assay, and we watch the speed with which yellowness evolves, and we extract the slope of that curve, and that gives us each data point in the graphs that I showed. That answer your question?
Yeah.
Okay. Yes, ma'am. So why only 4% ethanol? Well, eventually you'll kill them. Yeah, 10% kills them, and so we use well, actually our standard is 3% because it gives us a nice balance between retarding growth and stimulating a good response. So that's really the way we just discovered it empirically. With 1%, you barely get any response. With 2%, you get a small response. 3% works pretty great. 4 and above, you start slowing them. You and then you. The stress factors, so typically in the graphs that we show you, like the bulk assays, we do that experiment for usually 2 to 3 hours after adding the stress. And that's just because in a flask, eventually the cells stop growing, you know, they're just going to run out of nutrients. When I showed you the microfluidic data in the mother machine, once we switch to the medium containing the stressor, it stays in the stressor indefinitely. It's not just it's not a transient stressor. You switch into stressor and it stays in stressor. Make sense?
With a transient response.
They're still in stressor. Yes, yes, and you bring up an interesting point because you would expect that so somehow cells like adapt or they turn off, right? They respond and they stop despite the repeated exposure to stress. How that being turned off propagates generation after generation, we have no idea. Because we keep them in stress medium and we can watch them for tens of generations and we don't see another response. You know, after a couple of generations, all the proteins are new. And yet somehow they're synthesized in a state where they can't respond. That's something we still don't understand. Here, question is, are the stressosomes present all the time in the cell? Yes. Yeah. They're just there waiting until a cell encounters a stressor. So even unstressed cells have stressosomes there. And that's reflected by that. So the sigma A promoter, which is just a housekeeping promoter, which just means that it's on all the time at some level to produce the stressosome components. Yeah, and when there is a sigma B response, it's not making any more of the stressosome components, RAS and T. Rather, it's just making the downstream part of the parts that are involved in the stress response directly. No, there's no membrane bound nature or anything like that. Just a big protein complex sitting inside a cell.
of this, form these.
It's thought that they actually form as they're translated. So they're on one operon. So you have one mRNA, and so as the ribosomes are moving along that mRNA and these proteins are being made, they immediately bind to one another. That's the idea. You can like isolate them in vitro. Look at them finding them. You have a question?
Oh, I was just wondering, just like in general, this is a lot of work. I was wondering, like in the back of your head, what sort of applications would this like result in, like for like in the industry or like therapeutics or?
So there are pathogenic species that also use stressosomes, the most famous of which is Listeria. And so Listeria is a foodborne pathogen. So understanding more about sigma B could give us some groundwork to somehow figure out ways to make sure we have fewer Listeria outbreaks. But really, I mean, that's a little bit of a hand-wavy answer. We're really just studying fundamental bacterial biology, and the interest for us lies not so much in that application, but in studying the stressosome as like a molecular gearbox or a molecular machine as an example of what protein complexes can do in terms of taking an input and modifying the output. And so that might be interesting in biotechnology for making something that gives us a particular coded output to some input. Sure.
All right, with that, another round of applause for Dr. Khalid.
Thanks, folks on the Zoom.
That's nice. I thought you were-